It's a 13-year-long project which was completed on paper in 2003, and the sequence of chromosome 1 was completed only in May 2006, and it was funded by National Institute of Health, US Department of Energy, and Wellcome Trust, and it was later joined by Japan, France, Germany, and China. Then we discussed about the goals of Human Genome Project. The first one was to identify total number of genes in human genome. Second one, to determine the sequence of all 3 billion base pairs. Third one, to store the information in database. And fourth one, to improve or develop tools for data analysis. Fifth one, to transfer related technology with industries. And the sixth one, to address the ethical, social, and legal issues related to the project. Then we discussed about methodologies. There were 2 major approaches. First one, expressed sequence stacks. This approach mainly focused on identifying all the genes that can be expressed as RNAs. Then the second one, sequence annotation approach, also called blindfold approach. The blind approach is simply sequencing the whole set of genome containing both coding and non-coding region, and then later finding the function of it.
Yes.
Then for sequencing, the total DNA from a cell is first isolated and then converted into random smaller sizes, fragments of smaller sizes.
Yes.
Then the fragments were cloned in a suitable host using suitable host, mainly bacteria and yeast.
Yes, right. And then what after cloning?
And they were cloned in suitable hosts using specialized vectors. In bacteria, BAC, that is bacterial artificial chromosome, and in yeast, there's a YAC, that is yeast artificial chromosome.
Yes, these two are vectors, right?
Yes.
Just remember that these two here, this BAC and YAC, both are vectors.
Then the cloning resulted in an amplification of each piece of DNA fragment. Then the fragments were sequenced using Sanger sequencing. This sequences were then arranged on the basis of some overlapping region present in the sequence. Then this alignment was not possible manually. Specialized computer program were developed for this purpose. Then our Human Genome Project also led to rapid development of a new area in biology called bioinformatics.
Yes.
Then we discussed about the salient features of human genome. So should I read this?
Yes, you have to just tell us about each salient feature. Like what did we figure out? If you don't want to read it, you can just say that the first point we figured out that the genome contains this many bases, then this, just like what you did above for the methodology. Just like a quick summary of what were the salient features.
Yes, sir.
Yeah.
So first we figure out that the human genome contains approximately 3.1 billion base pairs. And the second one, that the average gene consists of 3,000 bases, but sizes vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. Third one, that the total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 140,000 genes.
Yes.
Fourth one, the functions were unknown for over 50% of the discovered genes. Then the fifth one, less than 2% of the genome codes for protein, that is cestrons. Then repeated sequences make a very large portion of human genome. Then repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. They are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, that is 2,968, and
Approximate 3,000 genes, yes.
And Y has the fewest, that is 231.
Yes, and all of them are genes. So remember, all of them are not protein coding genes, right? Because they are called cistrons. You understand? The region that codes for protein. There are genes that code for RNA but do not go on to form protein. Now I think this part is clear to you. Yes, and because we are talking about Y chromosome, something that I just remembered. So the biggest human gene or the largest human gene in terms of the length of base pairs is human dystrophin, okay, which is 2.4 million bases. You know which is the smallest human gene? That will also help you. Remember when we started reproductive system, I told you about the male and the female gender, that in nature, which is the default gender? What is the default state of a body in nature? Remember, I said that female gender is the default gender. And then to become male, you need Y chromosome, which has certain genes that in a specific timely manner tells the embryo to develop male gonads. Yes or no? Remember this?
Yes.
Everyone?
Yes, sir.
Yes. So this Y has the fewest gene, and also the smallest gene in human is also on the Y chromosome. And it's the gene that is responsible for maleness of the embryo, that makes an embryo male. And to make an embryo male, what do you need to develop? What do you need to determine in an embryo? If you don't do anything, the embryo will just develop ovary. But what do you need to determine if you want to make a male embryo? You need to determine testes, right? Am I not audible, or you are not giving these simple answers? Am I audible, people? Hello?
Yes, sir, you're audible.
Yeah, so you're not giving these simple answers. Okay. So to determine a male, you need to determine testes in an embryo, right? If testes starts developing, then we know that the embryo is going to be male. And the gene that determines testes is called TDF. TDF, or you call it testis determining factor. Okay. This is the smallest gene in human. Okay. It's present on the Y chromosome. Is it clear?
Yes.
And can you imagine how small it could be? See, the largest one is 2.4 million base pairs. So how the smallest gene, how small the smallest gene will be?
TDF, it will be this small.
Yeah, I understand. But how small? How many base pairs? Small.
Many small, very small.
Yeah, very means very is not a quantitative term. 2.4 million is a definitive number. 2.4 million base pairs is the biggest gene. So how small do you think is the smallest gene? It is just 14 base pairs. So 14 base pairs define testes. Now can you appreciate and understand why male is a non-default state and needs to be defined later on in evolution? You understood?
Yes, sir.
So it has only 14. So only in the whole genome. Now this is very, very interesting. When I got to know this, I felt that to be a male, You just need this 14 base pair long sequence in the DNA of the Y. Like you need the Y chromosome and you need this 14 base pair long gene that determines testes, and hence the male body is formed. Somehow rest everything is, you know, just present in the female body. The Y chromosome, these 231 genes, apart from these 231 genes, everything else is present in a female body. And these 231 genes are not very crucial to survive, to survival, to metabolism, or because it's obvious, right? Because Y is not present in females, and females survive. Not only survive, they give rise to new offsprings. So in evolution, the female gender is well equipped without these 231 genes. And even without this smallest gene, that this gene, testis determining factor, is called gene for maleness. So if majority of the male on the planet realize this, they will be very humble. You know, there's nothing to boast about. Maleness comes from 14 base pairs, but male ego is bigger than the whole genome. Fragile but bigger, right? Yes. So get rid of it when you are a biology student. That's my message to all the males here. And females, just respect these 14 base pairs. In evolution, you need 14 in 3.1 billion to be a male. So treat every male with respect. Male should believe that, you know, we just got here by luck. Goes both ways. Okay, anyway, so just remember this TDF because it has been asked once. It's not in NCERT, but because you knew the biggest, you should also know the smallest. Okay, so I think this is where we ended, right? And
Yes.
today, yes, so today we'll start the last topic of this chapter, and we'll wrap this chapter and start evolution. Evolution is, you're going to love evolution, all of you, I hope. Anyone who struggles in evolution, okay, we'll talk about evolution after we finish this. So the last topic is DNA fingerprinting. Now you know the meaning of these two words separately. You know what is DNA and what is a fingerprint, right? So what is a fingerprint? What do we mean by fingerprint? This, your biometrics, right? There is a very unique pattern.
Yeah, it's a unique biometric.
Yes. It can be used to identify a particular individual, right? Because
Yes,
it is very, very unique. So let me ask you a few questions. How unique it is? Like, will two identical twins have different fingerprints, or will they have same fingerprints? Two twins. Different? So they are made from the same zygote and still they will have different fingerprints? They will have everything same. Color of the hair, texture of the hair, eye color, nose shape, everything. They are identical twins. They both will be male or both will be females with almost every gene identical because they are actually formed from the same embryo which got divided into two due to some abnormality during division, cleavage division. So what do you think? Their fingerprints will be same or different?
Different.
Why? So you mean to say the moment you say that their fingerprint will be different, you are just saying that fingerprint is not genetically decided, right? It's not under the genetical control because everything that is genetically, because everything that is genetically controlled should be same in both identical twins, right? Their genetics is same. So if you say that their fingerprints will be different, it means that fingerprint is not controlled genetically. Yes.
Yes, sir.
Yes, so you are correct. Fingerprints. So how are fingerprints formed? What are these fingerprints basically? Look at your hands carefully, everyone. Look at the fingerprints in the thumb or in the top of your fingers. Look at a fingerprint. What do you think? How this formed? These formed during the embryonic development due to external factors, the pressure of the liquid in which the folds. So basically, if you see this, there are additional lines apart from prints, right? Do you see that in your fingers and hands?
Yes, sir, folds.
There are lines. So basically, these are those lines where you fold your fingers or hands, right? And these fingerprints develop due to the property of pressure and surface tension in the skin during the embryonic development. So it's all stochastic and random. So even two monozygotic twins, identical twins, will also lead to having different fingerprints. Okay, is it clear?
Yes.
Okay, so we know that fingerprint is something very unique. Now DNA fingerprinting, what do you mean by DNA fingerprinting? So we know that from Human Genome Project that most of the DNA is not coding for proteins, and most of the DNA is just the repetitive sequence. Look at the point 6. Repetitive sequence make up a very large portion of the human genome, right? And repetitive sequence are just stretches of DNA sequences that are just repeated many times, like 1,000 times. They just repeat one after the other. So it can be a very small one. Let's say this sequence, GACTACCG, So this is one sequence. It will just keep repeating 1,000 times. So GACTACCT, then again GACTACCT, again GACTACCT, and it will just 1,000 times keep repeating and make a bulk of the DNA. So that's called repetitive sequence. Okay. Now in that repetitive sequence, there are certain, if there is a mutation, so because they are not coding for proteins, right? They are not coding for genes, they are non-coding sequences. So if there is a mutation in a coding sequence, that will affect the gene, yes or no? May or may not affect the gene. But if there is a mutation in the non-coding sequence, will it affect anything in the body? No, it will not affect anything, right? Because it's not doing.
Because it's not coding.
Yeah, because it's not coding. So there is no consequence that will go down. So these single base pair changes are called SNPs. So in a human genome, there are about 1.4 million locations where single base DNA differences are present between humans. So that's called single nucleotide polymorphism. The name says single means one, nucleotide means at one nucleotide, which contains a base, polymorphism. So there can be many forms. It can be, so let's say single nucleotide morphism is like this, that at this place, in place of C, you have A. So it becomes GAATACCT, or you have G, it becomes GAGTACCT. You understand? Or it can be T. So at this one point, there are 4 possibilities. That is called polymorphism. Is it clear?
Yes, sir.
Everyone? Yes. So they are called SNPs. And this information of SNPs and Vntr, I gave you a homework to go back and read Vntr. Did you do that? What is a Vntr? Yes, no? Okay, so I'll tell you about this. Let's first write down a few things.
So
yes. Oh, Arpit is here, kya? No, it's who said sir?
sir, it was me.
Okay, Arpit is here.
Good evening, sir.
Yes, good evening. Why is Arpit pinned? Who pinned Arpit? Okay. Yes, Arpit, tell me.
So like what we have started.
Sorry?
So we have started with what, sir? Like what did you do?
We have started with
Before
DNA
that?
fingerprinting. We just started with DNA fingerprinting. Before that, I just told that the smallest gene is TDF, testis determining factor, which is just 14 base pairs long. Okay, it's the gene for maleness. That's all. Okay. Now, write down about DNA fingerprinting. This process involves identifying differences at some specific regions in the repetitive DNA sequence. In the repetitive DNA sequence. Now we know which regions are more susceptible or more vulnerable to these SNPs. So this has made our work easy. Suppose you want to distinguish between two DNA samples, that this sample belongs to one individual and the other belongs to other individual. So now you don't have to go and look at the whole genome. You understand? Like you don't have to do a whole genome sequencing to figure out the difference. You can just
Alright.
look at this localized region where which is more vulnerable to polymorphism, right? Then you see that one individual has this polymorphism, so the other individual, if it has a different morphic sequence there, then the two samples belong to two different individuals. If you collected a sample from a crime scene, let's say, and you want to figure out who's the criminal or whose sample does it belong to, so you take out suspect sample and just look at that region, and if it matches with any suspect, so that DNA from the crime scene belongs to that suspect. That's called forensics, DNA test, right? You must have heard of this thing, that through forensics you can get to know.
Yes, sir.
Yeah. So it's done through DNA fingerprinting. It's commonly known as DNA test. So you must have read or heard it from the perspective of testing for father or mother, biological parents, right?
Yes, sir.
Yeah.
Yes. So that's the same thing which we are studying right now. So how it works, let's see. Write down. Repetitive sequences, right on this point, repetitive sequences can be easily separated, can be easily distinguished or separated from the bulk genomic DNA. Okay, genomic DNA is very big, but it's very easy to identify a repetitive sequence, right? Because it will be repeating. So that's why it's easy, right? Yes or no?
Yes, sir.
Repeating means is a pattern, and it's easy to find a pattern in a scrambled, jumbled sequence, right? So it will be forming a pattern. So it is easy to distinguish or separate from the bulk genomic DNA through density gradient centrifugation. Through density gradient centrifugation. Okay, I found this. Through density gradient centrifugation. Okay.
So can you repeat the whole point?
Yes. Where did I start?
Through density gradient centrifugation.
No. Where did I start? He wants
Repetitive sequences can be easily.
Yes, repetitive sequences can be easily separated from the bulk genomic DNA. From the bulk genomic DNA through density gradient centrifugation. Okay. So the bulk will be the genomic DNA, right? And we will get other small peaks in this centrifugation, which will be called these, which will be forming these repetitive DNA. So this is known as satellite DNA. The repetitive DNA, which forms small peaks, small regions. Are called satellite DNA. You have studied about satellite DNA in class 11th during cell division and cell cycle. Remember, on a chromosome you used to make satellite DNA towards the very end, satellite chromosome. Remember that? No, it was in NCERT. Okay, anyways. So that's called satellite DNA. So either a satellite DNA is AT rich. Why it is AT rich? Because if it is repeating, then A and T will come multiple times. Wherever there will be A, there will be a T, right? A pairs with T.
Yes, sir.
So normally there are two type of chances. Either you can have a stretch of DNA that is AT rich, or a stretch of DNA which is GC rich. Okay. So we can easily identify AT rich regions or GC rich regions because of this repetitive property. Now, this satellite DNA, write down. So we have repetitive DNA, which forms the smaller peak, and it's different from the bulk DNA. So this smaller peak forms the satellite. It's called the satellite DNA. And satellite DNA can be of two types: microsatellite and mini satellite. It's on the basis of how long is the repetition. So micro and mini, both are small, but you know that micro is smaller than mini, right?
Yes, sir.
I got to know it through Uber cabs or Ola cabs. They give you a mic. Yeah, you too?
Yes,
good.
sir.
I got to know that micro cabs are compact, or those sharing one. Minis, then, so micro satellites have smaller repetitions, smaller sequences that repeat. Mini have slightly bigger, but that's not that you have to worry about. You have to know that both of them, whether it is micro or mini, basically the satellite DNA itself do not code for any protein ever. Okay, so write down, it does not code for any protein, but they form large portion of human genome. But they form large portion of human genome. Now, if there is a polymorphism, and yes, and these regions are the hotspots for polymorphism. Okay, so write down, these sequences show satellite DNA sequences. These sequences show a very high degree of polymorphism. Done. Things are sorted. We know which DNA to now target whenever you have to differentiate between two DNA samples, right? So the job is easy, right? No need to go for whole genome sequencing every time. And that's not feasible also economically. So since now we know these specific sites which show high degree of polymorphism, next time we will check these only. That's all. And we also know their locations on chromosomes. So it's easy, from a sample, just take out that chromosome and then you can make fragments of that sequence which shows polymorphism. Just sequence that fragment. You understand?
Yeah, sir.
Yeah. Okay. Now, write down the polymorphism. Just write down this one point so that whenever you revise, you understand. The polymorphism is simply variation. Polymorphism is simply variation at genetic level. Variation at the genetic level that arises due to mutations. Simple. So mutations happen, and these mutations do not have any consequence, so they stay there and then they are passed on. Is it clear? So that's how you can also figure out who's the father. Because if you have half of your genome from the father, you also have their polymorphisms, their mutations in these high degree of polymorphic regions, right? Which
Yes, sir.
someone else will not share. Okay, so that's how. Now write down. In other words, also write down one more definition. In other words, DNA polymorphism is an inheritable mutation. DNA polymorphism is an inheritable mutation which is present in a population in high frequency. Why it is at high frequency? Because it has no consequence. Any mutation, if it has a consequence, it will not be present in a high frequency because it will make the organism less fit or diseased or disordered. Yes. And even if it is making the organism better, that will also not be present in high frequency. Everyone will not become better. But things which are neutral, those things can be present in high frequency because no one cares. The organism does not care. The DNA, the gene does not care. Is it clear? Evolution does not care, basically. Is it clear to everyone?
Yes, sir.
Yes.
Okay. Now write down about the technique, the mechanism or the technique. Mechanism of DNA fingerprinting. How do we do it? First point, this technique was developed by Jeffries. Jeffries. He developed the technique. And what he did, next point, he used a satellite DNA. Now you know what is a satellite DNA? Small stretches of repeats that just small stretch of DNA that repeats over 1,000 times, right? So he used a satellite DNA as a probe. Now probe is something which can go and bind at some place. Okay, so that's probe. So he used satellite DNA as a probe that showed. So which satellite DNA he used? That shows high degree of polymorphism, of course. So you just have to identify a region that shows high degree of polymorphism and you can use that. And this making of probe, a satellite DNA as a probe, is known as Vntr. Because you can make it on your own. Like even if you go to research, you can make your own Vntr. Vntr stands for variable number of tandem repeats. And the name itself says variable number. So it can be any stretch that you take and make it as a probe and you call it Vntr. So Vntrs are the soul, the crux of DNA fingerprinting. We do it through this. So now how will you detect the Vntr? So we are testing which VNTR goes and bind where. Okay. So if the VNTR has been made, which is complementary to the polymorphic region of individual X. So you for that let's understand it like that. So there is a kid, and there are two people who are claiming to be the kid's father. Okay. So what you will do, you will take the kid's DNA sample. And how to take the DNA sample? From where we need, where is DNA?
In
How
the
can
cell,
so how can I take a DNA sample?
DNA extraction.
From where? Which part of the body?
Any part.
So just cut one finger off the baby. That is what I'm asking.
No,
So I
there's
have to,
Probably like a
cells.
cheek cell or something, right?
Yeah.
yes, any live cell, just take a toothpick, scrap the inner cheek, you will get lots of cell out that contains DNA. Or just take blood that contains DNA. White blood cells have nucleus, they have DNA. So you can take DNA from any part of the body, and whatever is the VNTR, if you have received it from your father, you received it in the form of a zygote, right? So every cell in the body will contain the same VNTR, sorry, the same satellite DNA, right? Whether it is the cell of the cheek, the bone, the blood, hair follicle. You can even take just a hair, like pluck it, don't cut it, and the base of the hair has this bulb. You know about that? So here has this one bulb at the base. It looks like this. Okay. So this bulb has some live cells. This also contains DNA. Is it clear? So this part is under the scalp and the hair is out. So you can take sample from here as well. So you take the sample, you look for the satellite DNA sequence, then you use that sequence as a probe, like make a complementary sequence that will be called a VNTR. Now you label that VNTR and make it radioactive. Okay. So VNTR is mostly radio labeled so that we can easily see. Okay. It's a radio labeled probe. Making sense to everyone?
No. Yes, sir.
Someone said no.
No, I
So
said
what
yes.
means radio label?
Yeah. Okay. You missed that class, Hershey and Chase experiment. Are you clear about Hershey and Chase experiment, who proved that DNA is the genetic material? Do you know that, Sahel?
Yeah, I've
Yeah.
seen that video.
Yeah. So they labeled. So radio labeling means you label something with a radioactive element. Now, in nature, there are natural radioactive elements, or you can make an element radioactive. So they use radioactive sulfur and radioactive phosphorus there to radio label DNA and protein separately. Similarly, you can either use some radioactive element or can make any element radioactive in a VNTR, because it's basically the sequence, right? So it's the DNA only. So you can make phosphorus radioactive and make a radio labeled probe. So radioactive things, they glow in the dark. You can see it on a gel. You can visualize the radioactivity. Radiums are radioactive in watch. Earlier watches used to come with these radium coated dials. Remember? So when you are in the daytime, they will absorb light energy, and in the night time they will radiate energy out and they will glow. You have not played with those radium balls or radium dial watches?
I got to know, sir. I understood.
You understood? Yes.
Yes, sir.
So we are
Yes,
just
sir.
making a radio labeled probe so that we can see that which sample it is going and binding to. So because we have made the probe from a satellite DNA of the kid, okay, kid's satellite sequence, repeating sequence. So whoever will be the true biological father will, when we allow these VNTR probes to go and bind to their regions, to whose DNA it will bind, to whose sequence it will bind, whoever is the father, right? You understand?
Yes.
Yeah. So on the gel, when you will get the bands, the bands will match with the biological father and not match with the one who's not the biological father. Okay, so you understood what is Vntr, how it is produced. Okay, so write down this technique. This technique includes, and this technique is also known as, earlier it was used with southern blotting. So write down the technique of radio labeled VNTRs involved southern blot hybridization. Hybridization is to bind to a stretch of DNA or RNA. Something which is a probe, it binds to a complementary sequence. That's called hybridization. And because this thing which is binding has its own detection thing, it is radio labeled. So wherever it is, it will bind, that region will also be, you can identify where is that region where it is binding. And because you know the sequence of the probe, you will get to know the sequence of the DNA strand it is binding to. So southern blot hybridization, there are two more, northern blot and western blot. There is no eastern blot. Southern blot was named after the scientist, okay, Edwin Southern. So his name was Edwin Southern. He figured out a hybridization technique to look for DNA. So it is a DNA detection technique. Write down southern blot hybridization. This is asked in entrance. Just because NCERT uses this word, you also have to know western and northern now, but just know what they do. Southern blot hybridization is to detect DNA sequences or fragments. Is it clear? Then there is northern blot. Now, northern and western were not named after scientists. It was just to keep the, it's interesting, keep it similar to southern blot. So southern was a name, but it happened to be a direction also, right? Southern direction. Yeah. So when they developed a technique, so similar kind of hybridization technique to trace and detect RNA, they call it northern blot. So south and north, DNA is south, then RNA is opposite north. And can you tell me what is western blot? What is the other thing that you need to detect in a cell? DNA, RNA, and what is the third thing in the central dogma?
Protein.
Protein. Yes. So western blot is for protein. Now, will you remember it now?
But so why west, not east?
Yeah, probably because the Western countries developed it.
Yeah, that's all smart. Yeah.
I don't know. Like that's not. It just, yeah, probably because of that, right? Because
Probably.
we call America as West, right? And we call Japan and India as East. So whatever Western culture, so Western blotting, probably.
Probably.
And because there is no fourth thing to detect, so there's no Eastern blotting. DNA, RNA, and protein. So southern, northern, and western blot. So this technique was used along with this southern blot to figure out DNA sequence. Now, how does the technique work? Write down steps of DNA fingerprinting. First, again, anything you have to do with DNA or RNA, you have to isolate it from the cell. So first is isolation of DNA. Okay, first 2-3 steps are same like human genome. There you have to sequence the whole genome. Here you just have to know the difference between two polymorphic regions. That's all. So first is isolation. Then what will you do after isolating? You will make fragments, because DNA is very big to do anything, right? As a molecule. So you have to, whenever we have to work on DNA, we have to make fragments, like any sequencing thing or identification thing of the sequence fragment. So this time In Human Genome Project, what was the fragment? It was random. You make random fragments, right? So that you can then annotate it. You can do sequence annotation and can again align it in the proper sequence. Yes or no? Do we have to align it here? No. And we also know which region we are interested in. So we will not make random fragments here. We will make targeted fragments. So write down targeted fragments, which is also known as digestion of DNA. Digestion of DNA by using what will be the enzyme called that digests DNA? DNA is a nucleic acid. So what will it be called? DNase or else other name. So ones that digest proteins are called protease. And if I say that specifically, that yes. So digestion of DNA by the enzyme called RE, restriction endonuclease. Now this is the enzyme and it's called endonuclease because it cuts the DNA from within. So there are two types. Let me explain it to you. So one is endonuclease. Let's say this is a DNA fragment. So endonuclease will cut it from the center anywhere, right? This fragment will be cut out. Is it clear? Then there is exonuclease. And you can understand in exonuclease, if this is the DNA, it starts cutting the DNA from the side, from outside. It cannot cut from inside. Do you understand? If something is digesting DNA from outside, from the ends, it's called exonuclease. And if something can cut anywhere in a targeted manner, wherever you want it, that's called endonuclease. Is it clear, everyone?
Yes, sir.
Yeah, sir.
Yeah. So this fragmentation is through restriction endonuclease because we know what fragment we are interested in. Now you have fragmented. So third thing, what you will do? You will separate the fragments, right? So that's called separation. Separation of DNA fragments by a technique called electrophoresis. Again, don't get intimidated by the names. Restriction means that restrict the size. Endonuclease means that cuts. So you cut and restrict a size. Suppose you designed a restriction endonuclease to cut the DNA from one point to other, carrying 40 base pairs in between. So you are actually making 40 base pair long fragments. You can make 100 base pair long fragments. You can make 200 base pair long fragments. So you are restricting the size through endonuclease. It's called restriction endonuclease. Simple. Electrophoresis. You are using electricity to do separation. Okay. So electrophoresis, it can be done on DNA because DNA as a molecule is negatively charged. Correct? It's all just logic. Do not mug up biology. So DNA has negative charge. So if I make random fragments of DNA, so let's say these are the fragments of DNA which I made. All are DNA fragments. So all will be negatively charged, right? Some fragments are bigger, some fragments are smaller. Now what I do, I apply electric voltage. So these fragments, there will be a gel. So there is a gel.
Agar gel.
Yes, agarose gel, which has these wells, place where you can load the sample. And then from one side, now DNA is negative charged. So you will apply negative charge here. Sorry. You will apply negative charge here and positive charge here, right?
Yes, sir.
In a circuit. containing, let's say like this. Now, because DNA is positive, negatively charged, it will move away from the negative terminal because negative repel, and it will move towards the positive, right? This will be the direction of movement. Now, because this agarose gel has a pore size, you can make gels of different pore size through different concentration of the material. So if you make highly concentrated gel, it will have smaller pore size. If you make loose gel, it will have greater pore size. Now, depending on what fragments you are making through endonuclease, you make a gel, 1% gel, 2%, 3%, like that. And then you are allowing the DNA to move. Now, the DNA fragments, which will be smaller, will move faster, right?
Yeah.
They will move easily, and they will be present. They will show a band here. You understand? Suppose this is well 1, so there is a band here. In well 2, there is a band here. So what can you say? We can say that this DNA was smaller in size than this DNA, right?
Yes, sir.
So similarly, you can make a comparative analysis. You understand? Is it clear?
Yes, sir.
Sir, and
Yeah.
also like they are less negatively charged. So,
Sorry, what?
so like it can also be like a possibility that the smaller ones are less negatively charged and the bigger ones have more charges. So, would it be a
So?
reason? It is.
See, both are negatively charged. There is nothing like so. Less negative charge and more negative charge works when the bodies are of same size. When the body is smaller, if you have 1 kg mass versus 10 kg mass, and on 1 kg mass you have 1 unit of negative charge, on 10 kg mass you have 10 units of negative charge. Basically, it's the same. No? It is 1 unit of negative charge per kg. You understand?
Yes.
So the negative charge is coming from the phosphate group. So the longer the DNA, more phosphate groups. But again, it's longer, no? Smaller the DNA, even if there is 1 phosphate group, it is 1 base pair long DNA, it will be attracted toward the positive terminal, and it will move faster because it can move through the pores without resistance. You understand?
Yes.
So basically you are pushing all the DNA through a pore size. Now the smaller one will move faster and the bigger one will move slower because they will be in a traffic jam.
Yes.
Make sense, everyone?
Yes, I got it.
Very good. So that's called separation of DNA. Once you have separated the DNA, that's not done. After separation, you have to, why it's called a blotting technique, because we cannot visualize here what is the band. So you have to transfer. So after separation,
Stain.
Sorry?
The staining.
No, first transferring, then we will stain. So this gel will be kept close. It's just like, suppose if I make color, if I apply some color, I make a pattern with and you must have done it, right? Have you done that on erasers? When I was in school, I used to do, so on erasers, we make something with a pen, then you press the eraser against your skin, and you're like, we got a tattoo or a stamp, right? Fatima?
Yeah.
So the eraser is like the gel membrane, and our hand works as another membrane on which we can just transfer the impression. So the 4th step is transferring or blotting. That's why it's called blotting. You know about blotting paper? Why I'm writing paper? So this process is called blotting. That's why it's called southern blotting. We are blotting for DNA. So transferring of separated DNA fragments. Write down. Separated DNA fragments to synthetic membranes. To synthetic membranes. The membranes that are used are nowadays nitrocellulose is used, or nylon membrane is used. These 2 membranes are used for transferring. Once you have transferred the same impression, so and you do the same thing, there's no rocket science. You just take the gel, take the membrane, keep it close, and then cover it with tissue paper and apply weight from outside. So they're literally like in my lab, one of my seniors used to do Western blotting, and when he used to do transfer, he literally used to keep books as weights. So big books you can keep on the top so that it stays for a few hours under constant pressure or weight, so that the transfer can happen very effectively. And that's how you transfer basically. Okay, just like we do for stamp, we press it so that the impression gets on the other surface. Yes or no?
Yes, sir.
So we did that. Now, after we have transferred what Arpit said, what do you say? Labeling? Staining.
So staining.
So here we are basically just hybridizing. We don't use. Why does Siri gets activated all of a sudden?
Sorry, I'm still not.
It's okay, Siri. It's okay. Okay. So what was I saying?
Sir, staining.
Staining. Yes. So once we transferred, now we will do hybridization, because we don't need stain. We have already Vntr probes ready. Remember?
Yes, sir.
So we will just put those probes and see where it goes and binds. Okay. So that's called hybridization. Hybridization using labeled Vntr probes, because they are already radio labeled. So wherever they will go and bind, we will know that yes, this fragment of DNA is present in this sample, and it's also present in this sample. So both these samples are coming from common source, which means he is the father and he is the son, and it's not matching to this sample. So this person is not the biological father. Okay. Easy or difficult?
Easy.
The concept is easy, right? The concept is very logical. Science is very logical. You understand it, it's easy. Only when you do it, it requires some expertise. That's a different thing. But the logic is clear to all of you.
Yes, sir.
So we have labeled through hybridization, kind of we have stained, what Arpit said in layman's term, and then we need to do detection. So detection of hybridized DNA fragments. Okay. And how do we detect radio labeled things? They emit fluorescence, like they emit energy in a wavelength which we can see. So that's called, the technique by which we detect radio labeled probes is called autoradiography. The terms, again, let's come to the term. Auto means
Automatic.
self.
Sorry, sir.
Auto means self, right? Automatic, self-working machine, right? So auto means self. Radio means that radio labeled probe. Graphy means we are just detecting it. So we are just detecting the automatic fluorescence that molecule is throwing out, the light. So from the radium, automatic lights comes out, right? It emits. We just have to see it, detect it. That's all. So it's called autoradiography. Is the technique clear to you logically?
Yes, sir.
It was not at all difficult. Let me just, if you have NCERT, please open your NCERT, everyone. NCERT, and can someone tell me the page? There is a diagram for DNA fingerprinting.
122, sir.
123.
Sorry,
123.
123.
Yes.
I had 122 also. Is it different in different books?
Oh, no, sir. I thought you were going to ask about the steps, so then I said 122.
Oh, there are steps also. Okay, good. Is there anything I missed? Please, can you confirm me, Matthew?
No, sir. All 6 steps are there.
Okay,
So-
very good. Yes, so there is 1 minute. I'm pulling out this figure, Arpit.
Sorry.
Then you, no, you ask the question. Let me just, oh yes, this one. Okay. Command 4. And, yes. Yes, Arpit, tell me.
Sir, a question came in a test say that was like we have to tell the applications of DNA fingerprints.
Mhm.
So, sir, can you tell?
You tell, no? I told you what is DNA fingerprinting. Now I gave you, if I gave you a mobile phone, then you are asking what can I do with this? You tell me what can you do? You
So
know that we have a
it-
technique through which we can distinguish between two different samples of DNA. Not you tell me.
say like it can be used in the criminal scenes for forensics.
You see, so you already know. You know everything, Arpit. You do not forget that you scored full marks. I never scored full in my biology exams ever
Say that the chapters were easy.
because you knew it, right? Just 1 second. What I'm doing. Can you see this figure, everyone?
Yeah, sir.
Yes, sir.
Yeah. But I'm happy that my students are doing something that I didn't do. So I'm at least happy that you will do better than me in biology. Expecting some good doctors and good scientists coming up. Okay, now look at this figure. Let's try to see what's happening. So this is a DNA from crime scene, DNA C. Okay, let's first look at this DNA because this came from a crime scene. So you are an forensics expert and you were told that there was a person murdered, and while he got murdered, he also struggled and also had got in a fight with whoever killed the person. So there are two type of blood samples present on the scene. One sample belongs to the victim who's dead. The other sample does not matches to the victim. So whose it can be?
Killer.
The murderer. You know,
Yes.
especially if it is present. So sometimes when you fight, you might get like you are trying to defend yourself, and some tissues, like if you scratch or if you try to get rid of the person, some tissues might get stuck in your nails, right? In the victim's nails. So you can do a nail swab, and if you see a DNA in that nail which is not the victim's DNA, so clearly he was in a fight with someone and he scratched someone and got some tissues, right?
Yes, sir. Also in like a very hypothetical issue, like situation, like if the person is using knives, he can probably cut himself and have blood on him.
Yes, right. So on if on a knife you find two type of bloods, then again, so inside the blood or from inside the tissue you can get some cells and you can isolate DNA. You can do all the 6 steps that I told you, and they figured out that this DNA from the crime scene had some VNTRs that were detected with different varying lengths. So from length 0 to 11 repeating sequence or less than 11 on chromosome number 7, chromosome number 2, and chromosome number 16. These were some unique regions which were having these VNTR repeats, and they are more prone to polymorphism. We know that polymorphic sequences, satellite DNA. Now there are two people, individual A and B, who are suspects. So you did the same thing. Now again, every chromosome has 2 copies. Can you see that? And why is the VNTR different on 2 chromosomes from the same sample? Because one chromosome is maternal and the other chromosome is
Paternal.
paternal, right? So in this case, because it's from a crime scene, both the chromosomes will match, right? Because it's the forensic being done for finding the criminal. Now, can you tell me which is the criminal, A or B? So this A date individual's gel is this. This is A's data, this is B, and this is the C's repeat on a DNA gel. Which one do you think is the criminal? B, right?
Yes, sir. B.
But note here that some VNTRs, let's say this one, this actually also matches with A, right? In fact, this one, blue one, smaller one, this also matches with B, A here.
Yes,
See
sir,
or
but
not?
there's more similarity with that of B.
Yeah. So we need more regions. If someone just took chromosome number 7 and 2 and this region, then what will happen? You may convict wrongly A also, right? So you need to see that multiple regions have the same similarity. See, this is exactly same as B, right? But with A, it's matching somewhere, not entirely, and it should entirely match. If it is the sequence, the satellite DNA of the person, it should match exactly, not just at 1 or 2 sequences, because that is the power of polymorphism. You can just have multiple polymorphic regions in an individual without any consequence. You understood, everyone?
Yeah, sir.
Yes.
Now let me just confuse you a little bit and ask you, in case of a paternity test, let's say you're testing for a biological father, what will be the difference in the detection? Apply your brain and tell me. If it is not a forensic criminal matching case, if it is paternity case, okay, we know who's the mother. The question is, who's the father? Mother is saying that he's the father, but the father is saying, no, I'm not the father. Then there's a test done with the kid and the father. So what will be the difference? It is not for the same individual now, right? It is for
Yeah, sir.
paternity
It's a
test.
mix of the
Most of
mom
them will
and the
be
dad.
matching.
Sorry, most of them will be matching.
Yeah, and
No,
some
other
of
than the mothers, it must match, right?
So exactly half of them will match, because rest half will be coming from mother, not most, not few. So for every chromosome, one strand will be matching. In this case, you see both the strands are matching. Can you see? This and this is matching with this and this. Yellow both are matching with both yellow. Both blue are matching with both blue. Because this sample is from this individual. Same. So both mother and father will be same. But for a kid who is being tested, half will be from mother and half will be from father, right? So when you're doing the paternity test, so this kid's father also had his father and his mother, right or no?
Yes.
But he only contributed half, right? Half to this kid. So only the father's regions will match exactly. The mother's region will match with the mother exactly. So you can tell who's the mother and who's the father. You understand? So for a paternity test, you will see exact half of the strands, one strand in each chromosome matching. Do you understand, Fatima? Not few, not some, not most, exact half.
yes, sir.
Yes? Clear,
Yeah.
Matthew?
Yeah, sir.
Okay, very well. So with this, we come to the end of the chapter. And before I start the basis, like I'll introduce a little bit of evolution. At least I'll prime you to that. I'm giving you 3 minutes, everyone, to revise the whole chapter. Go through the whole notes and ask me doubts if you have any doubts. And even those, Sahel, you have any doubt with this chapter? Samaira, any doubt? I am taking the doubt for this chapter now. Take 3 minutes, go through the whole notes right now. Okay, I'll be back with some water. Oh, I have the bottle here only. Okay. Just are you revising, people? Say yes or no, in fact.
Yes, sir. Yeah.
Okay, just 1 second. Any doubts? Anyone? Okay, take your 3 minutes. Let me know when you're done.
So,
Yes, Arpit.
so can you tell what is the difference between the clover leaf and inverted L structure? What is the basis to
Okay.
distinguish them?
You're asking about the tRNA, right?
Yeah.
See, it's simple. So the actual 3-dimensional structure, okay, which is the inside a cell, everything is 3D, right?
Yes, sir.
Yes or no?
Yes, sir.
So so imagine you're looking at this bottle,
Yes.
and currently you're looking at it in a 2-dimensional space, right? On a screen. So what is the shape of the bottle?
Rectangular.
Yes, it is rectangular, right?
Yes, sir.
But is it actually rectangular?
So it's cylindrical.
It's cylindrical. Exactly. So this is the difference. When you look at a 3-dimensional structure of tRNA, it is like a clove leaf. You know what is a clove?
Yes, yes, sir.
Yes, very good. So it looks like that clove leaf, but it's a little bent towards one side. So in a 2D, when you look at it, it looks like an inverted L like this. So the structure is something like this. Okay. More one bulges towards one side. You understand?
Yes, sir.
When you look at in a 3D, and because this bulge is because here is amino acid binding site. So when you look at this structure from in a 2D, does it look like an inverted L to you?
Yes, yes, sir.
Yes. But
It is.
imagine this structure in 3D, so you can rotate it, so it will look like a clove leaf. Okay,
Yes, sir. Okay.
that's the difference. Any other doubt, people? So the technicality of both Human Genome Project and DNA fingerprinting can be asked in entrances in greater detail. So that's why I spent some time and explained the mechanism to you. For boards, you just need to know the steps, the underlying principle, and the logic. What are we doing? Okay, methodology basically.
So,
Yes, Arpit.
sir, did you appear for NEET exam?
Yes, I did. Like when I was, I think at that point of time, you are not very sure what you want to do. So it's just I think it's a culture. If you have biology, everyone, almost everyone, those who do not, I think either they are very sorted, but I thought of doing MBBS once. And ultimately, I wanted to go into research. So my plan was to do an MBBS, become a doctor, and go into research as a doctor, because many people do that. I have seen people going into PhD research after becoming a doctor. So earlier, I wanted to work on diseases. And specifically, as a kid, I was very fascinated by neurodegenerative diseases, dementia, Alzheimer's, things like that. So I gave NEET. And I want to know, did I qualify it or not?
Yes.
I qualified, but I was not getting a good economical government college. So I then changed course. I had a plan B, because I cannot give so much money to go and study MBBS. So let me go through a BSc and a MSc and then go to PhD. You have to change course sometimes, right? So the qualification criteria of NEET is not that hard. I think it's not difficult to qualify NEET. What's difficult is to get into the ranks where you get the college you want, right?
Yes.
Yeah. And when I was in school, for me, like I'm very honestly, I was not very good at understanding the things that I'm teaching you today. So no one taught me DNA fingerprinting in this like this much of detail that I could understand the way I understand it now. And I see many of you, way ahead at your own levels when I was in school. So I'm very hopeful and very, very happy for all of you that you understand biology and you understand with some zeal and passion and some dedication. I was not very good at subjects, though I used to come first in the class, but everyone was like me only. Okay, any other doubts? But I'm happy that like I'm doing what I wanted to do, getting into neuroscience research, living the life I always wanted to live. So I'm happy. Teaching biology to kids who are brighter than me at in the age. Okay, anything else you want to ask? Matthew, Sahel, Samaira, Fatima? No one asked any doubt. So I'm assuming that you are clear with this chapter.
Yes, sir. I would say most of the doubts are set already. So.
Okay, done already. Okay, so we don't have much time to start evolution experiment, but okay, let me tell you one thing here only. I'm giving you something to think. We are going to read the next chapter, which is evolution, or biological evolution being the focus. Now, evolution is the most misunderstood concept in biology, not just among the common masses, but also among students and teachers, believe me. You all must have heard some loose statements, but you must have never read it in any book. If any book has written these statements, that book is wrong. But have you not heard of statements or pictorial diagrams where they suggest that humans evolved from monkeys or apes?
Yes.
Yes.
Yeah, definitely, sir. Yeah.
That's we think evolution says that humans evolved from monkeys, right? That's a misconception. That's a misconception because what evolution says, you never studied or understood in detail. You just, take away message. It's like a Chinese whisper. You say something in someone's ear without context and he will say something else. So the number of mouths it will go through, it will change. Information will change, right? So that's happened with evolution as well, because it was easy to understand, but it was difficult to contemplate in its details. So the statement that humans evolved from monkeys is not correct, because we still have monkeys right around us. If humans evolved from monkeys, then all the monkeys should someday become human. Yes or no?
Yes, sir.
Why are there still monkeys? Why have not all the monkeys become human? And similarly, monkeys evolved from something else. So why are there those organisms still? So that's not true. So the approach that I take to teach evolution, because evolution, genetics, and neuroscience are my core favorite areas in biology, that gives us answer about our own existence, about human culture, about human evolution. So the way I'm going to teach you is, whatever you have learned about evolution, you have to first unlearn it. Okay, so in the next class, when you come with a blank slate with no preconceived notions of evolution. No one will from now onwards believe that humans evolved from monkeys, because we do not. Then what happened? That's what I'm going to teach you. Okay, so everything that you know about evolution, forget it. Done? Promise?
Yeah, sure, sir.
If someone asks what is evolution, we don't know. Okay, we'll start thinking from the scratch. How life evolved. So there are many theories regarding it. The theory of abiogenesis. Another misunderstood statement is that non-living things became living in evolution. So I was having this one discussion with one of the friend that there has to have a special design, creating design. Someone has to design all these things. But according to science, there is no design that's needed. And I'll tell you why there is no design that's needed. Why the best that suits the environment gets selected over time with some evidences. Okay. In science, we never believe whoever say something that is said by someone, whoever that person is. Okay. Even even the things that Stephen Hawking and Einstein said are being challenged to be proven right or wrong. It's not just because Einstein said it, it must be true because he was Einstein. No. In science, everyone is student. Someone is senior, you can be a junior, 20 years junior in the field, but still you are a scientist. You can challenge something if it does not seems logical to you. So the theory of evolution over time got reshaped and changed and fine-tuned with evidences that emerged. When Darwin was there, there was not enough knowledge about fossils. Okay, then when fossils evidence started coming up, we changed a little bit. Let's see, these things are now well understood, so this must be happening. There are experiments done in space to show that non-living entities have this property of self-arrangement or self-alignment. So you all know about lipid bilayer, which is our plasma membrane, right?
Yes, sir.
So plasma membrane is lipid bilayer, right?
Yes, sir.
Now I just want you to go back to your class 11th and revise cell. Will you do it for me?
Yes,
in
sir.
this week? Revise cell. What is a cell? What are the various structures present in a cell? What makes a bacteria, which is a single cell, an organism? So these things you have to just brush up. Second thing that you have to brush up is natural versus artificial selection. What is this? What do we mean by natural selection and artificial selection? And third thing that you have to read, find out and read from your NCERT. Theory of chemical evolution. In evolution chapter, we are going to use some concepts of chemistry as well. So you have to just find theory of chemical evolution, and it is known also by this name, abiogenesis. Abio means non-living, bio means living, a means non-living genesis, abiogenesis. So we'll start from these basic things, okay? By the end of this chapter, my motive is that you, all of you, understand the concept of evolution in a correct sense. Okay?
Yes, sir.
Okay, so we'll start evolution in the next class. Please come prepared and please forget all the preconceived notions. And this is very, very important. I want everyone to contribute. Okay? Because see, I'll be very honestly, many a times people do not believe in evolution because they do not understand it. They think that it is a contradictory theory to our theological, religious, and cultural beliefs. But believe me, it's not. It's the most logical concept in science that you will ever come across. Okay? So let's take it like that. Let's understand it. And you have all the rights to ask questions. Whatever you do not believe in, ask. Say, I don't think this can happen. Can you prove how this happened? Proof. Ask for proofs, not just for the words written in NCERT. Okay? And to ask for proofs, you have to know what are few things. So come prepared, read these few things, and then come. Okay? Okay, everyone.